Welcome back, my dear students. Now in this session I'll be talking about a miscellaneous topic, you can say, that is free radicals and antioxidants. Now why did I start it now is the time when you have already finished electron transport chain. So this might be the time when you come to know what is exactly happening in free radicals. So some basic redox terminologies. I want you to look at the slide. Oxidation is gain in oxygen, loss of hydrogen or loss of electrons. Reduction is loss of oxygen, gain of hydrogen, gain of electrons. These terms have to be known to anybody. Now oxidant is oxidizes another chemical by taking electrons, hydrogen, or by adding oxygen. Reductant reduces another substance by supplying electrons, hydrogen, or by removing oxygen. These are the redox terminologies. Now with this background, let us move to what are free radicals. Now free radicals means those substances or a molecule or an atom or a molecular species capable of independent existence, that is by itself it is capable of staying, but it possesses one or more unpaired electrons in the outermost orbital. Basically we say one or more than one unpaired electron. Unpaired electron is a key word here. Unpaired electron in the outermost orbital. So you might have seen many of the times we put in many ions we put the negative charge, but when it is an unpaired electron we put a superscript dot also above it. So it might be having a negative charge or not, but if it is having an unpaired electron in it, we write it as a superscript dot over it. So in the outermost orbital it should be having an unpaired electron. So they are usually represented as R dot, and when are they released? Remember, complete reduction of oxygen results in the formation of water. So incomplete reduction, suppose there is partial reduction of oxygen, it will result in the formation of what are called as reactive oxygen species, also called as ROS. If you get the similar incomplete reduction from nitrogen, we call them as reactive nitrogen species or RNS. So ROS and RNS. Remember, in this they need not be free radicals also, they may be just be reactive oxygen species. They will come under the category ROS also. So now let us go further. Which are the reactive oxygen species? Some of them are radicals, some of them are non-radicals. So let us look at it. Superoxide anion, hydrogen peroxide. Hydrogen peroxide is a non-radical, but it is a reactive oxygen species. Hydroxyl radical, hydroperoxy radical, lipid peroxyl radical, hypochlorous acid, ozone, singlet oxygen, etc. are the reactive oxygen species. So this whole thing, these species are called as reactive oxygen species. Now let us go on to RNS, reactive nitrogen species. We include nitric oxide, free radical, nitrogen dioxide, peroxynitrides, alkyl peroxynitrides. I have just shown the symbol in each of these, but you did not remember any of them. So what are the characteristic features of these reactive oxygen species or RNS are that they are extremely reactive. Within a 30 Armstrong diameter of the formation of reactive oxygen species, it can react with anything and everything. Lifespan is very short. Within half milliseconds or nanoseconds, they are completely neutralized. They generate new reactive species by chain reaction and they cause damage to the tissues. These are the characteristic features. Now, how are they generated and when are they generated? So, for the reaction of ROS or RNS, we have endogenous sources and exogenous sources. I have just listed it out here. The endogenous generation of ROS, one most important is the electron transport chain, phagocytosis, reaction with metal ion or transition metals, xanthine oxidase, arachidonate pathway, lipooxygenase pathway in the platelets and WBCs, peroxisomes, exercise, inflammation, ischemia, reperfusion. There are so many causes in wherein the reactive oxygen species are generated. Now some of them you can see are enzymes, some of them are pathways, some of them are physiological states during which reactive oxygen species are liberated. So we go on to the important ones, the electron transport chain and leaks from the ETC. So I have shown it here schematically. Please note that. So as I said, partial reduction of oxygen. Complete reduction of oxygen will result in the formation of water. So in between that, partial reduction. Suppose oxygen takes up one molecule of one electron. What will happen? It will get converted to superoxide, O2 minus, which will again take up electron or proton. It will result in the formation of hydrogen peroxide, which can again react with the formation of water or hydroxyl radical, and this can again take up electron to form water. So in between all this, so from the formation of oxygen to water, there are a lot of times so many radicals are formed. All this can leak out from the electron transport chain. It is very easy to say that in complex 4, electrons are given to oxygen to form water, but actually it is not so. It takes place bit by bit. One electron goes, then one proton goes, then some 2 protons go, and thereby from at complex 4, water is released. So during this time there may be leaks that may be happening in the ETC, and all these leaks will result in the release of reactive oxygen species. Now, one more important mechanism whereby reactive oxygen species are happening within the cells is especially during the respiratory burst, which is happening in macrophages to kill the bacteria. We have already seen that, but I'll just revise it. So glucose forms NADPH by the HMP shunt. Now NADPH is acted upon by the enzyme NADPH oxidase, and it reacts with, it gets oxidized by oxygen to form superoxide. This is NADPH oxidase. Superoxide is further converted by superoxide dismutase to form hydrogen peroxide. Hydrogen peroxide is converted by myeloperoxidase to form hypochlorous acid. All 3 of them, that is superoxide, hydrogen peroxide or hypochlorous acid, all 3 of them are responsible for the killing of the bacteria in macrophages, especially in the phagolysosome. This sudden increase, when all these are being formed, there is a sudden increase in the consumption of oxygen by a macrophage, and this is called as the respiratory burst that is happening. So during this, killing of bacteria is something which every macrophage has to do. Whatever bacteria has been opsonized, it has to kill it. So during this, all these have to be formed, but they may leak out. That is the main problem with this. So these reactive oxygen species may leak out. Now, other important mechanism by which by itself these ROS can be generated is by reaction with iron or other transition metal. Now, 2 important reactions are there in this. I just listed it down MCQ point of view. One is the Fenton reaction and the other is the Haber-Weiss reaction. Please look at the slide. Ferrous iron reacts with hydrogen peroxide to form ferric plus hydroxyl radical. Remember, once an hydroxyl radical is formed, it can keep on generating more and more hydroxyl radicals. It can keep on generating more and more, not only hydroxyl radical, all the reactive oxygen species show the same characteristics. They can propagate. So, Fenton reaction, once it is formed, they can keep on propagating hydroxyl radical or hydroxyl OH minus. Now there is one more, Haber-Weiss reaction, in which oxygen combines with hydrogen peroxide to form O2 plus hydroxyl radical and OH minus. So again in this, once a free radical is formed or a reactive oxygen species is formed, it can keep on progressing. So this is the generation of reactive oxygen species. Now let us go on to other causes. We now saw the 3 important endogenous causes for the formation of reactive oxygen species. What are the exogenous causes? Are there any other reasons by when reactive oxygen species are formed? The exogenous causes include ionizing radiations, UV light, cigarette smoke, carbon tetrachloride, pollutants, drugs, pesticides. All these can result in the release of reactive oxygen species. So, are there any physiological advantage of releasing these free radicals? Yes, so what is the role of free radicals? One of the most thing is that if you look at nitric oxide, it is a smooth muscle relaxant. So like that, all these have got a particular role, and that is why they are generated within the body. So we have smooth muscle relaxation, neurotransmission, immune regulation, helpful in phagocytosis by destroying invading microorganisms. So they are beneficial to the body, and that is why they are released. So when are they not beneficial to the body? What will happen to them later on? What is happening to all these free radicals? So let us look at the effects of free radicals. So free radicals basically damage the cellular integrity. They can damage a cell. They can cause cell death. They can damage protein. They can damage DNA. They can damage carbohydrate. They can damage each and everything within its vicinity. They are that reactive. They can damage anything and everything. So they damage the cellular integrity due to its high reactivity. They react with all the biomolecules present and thus affect their normal function. Normally what it should do, it should go and react, do its function what it is supposed to do. But because of its reactivity, not only that, they will do something else also. So they can react with all the biomolecules present. So let us look at the first biomolecule with which free radicals can react, and that is lipid peroxidation. What is lipid peroxidation and how is it important? Now in all this lipid peroxidation, what is happening? The free radicals are reacting with lipids. And where are the lipids present? They are present in biomembranes. So how do they react? Basically it is considered as three phases: initiation, propagation, termination. Please look here, the reactions I have put down here. Nobody need to know about the reactions. Just I have shown you how a free radical reacts with a lipid and it is converted to lipid peroxides. They are converted to another free radical. Lipid peroxides by themselves become reactive. And this is maybe because of metal ions, they maybe have started, some initiation has started because of that. Now this is further what happens, they react with oxygen, form more free radicals, and these will again react with one more and keep on propagating. So initiation may require a metal ion, later on it doesn't require. This we call it as the death kiss of free radicals. One free radical will act with one more compound and make it a free radical, that free radical with one more to make free radical. So they will multiply and multiply. So this is known as the death kiss of free radicals. So this is the initiation phase and this is the propagation phase. But can it terminate? Yes, it can terminate. There is a mechanism by which it can terminate, and this is known as the termination phase. Now in this, please look that two free radicals will react with each other to form a non-radical product. Two free radicals combine to form a non-radical product. So all these non-radicals, we call them as lipid radicals. So lipid peroxidation has got terminated, peroxidation has occurred and it has terminated. It will result in the formation of a lipid non-radical. Now we can estimate this radical and it will tell how much of lipid peroxidation had occurred. So we can come to know what is the level of free radicals or the level of damage to the lipid layer. So this degradation of lipid radicals leads to a formation of a non-radical known as malone dialdehyde, MDA, and this can be used as a measure for lipid peroxidation. Now, what will be the consequences? Lipids have got peroxide. It has initiated, it has progressed, propagated, and it has terminated. What will be the consequences? Basically, it results in structural changes in the membrane. It alters the fluidity. It alters the activity of different channels. They alter membrane-bound signaling proteins. It increases ion permeability. So, and plus these lipid peroxidation products can form adducts or cross-links with non-lipids like proteins and DNA. So it can lead to a lot of consequences. Now let us look at the consequence of free radicals on carbohydrates. So hydroxyl radicals can react with carbohydrates. We saw how the free radicals react with lipids. So let us look at the effect on carbohydrates. When they react with carbohydrates, they produce a carbon-centered radical, and this leads to chain breaks in very big molecules like hyaluronic acid. So it results in a significant decrease in the synovial fluid in the case of especially the affected joints, and it is implicated in arthritis. Now we go ahead with further effects. What is the effect of free radicals or reactive oxygen species on amino acids of protein? So proteins may contain proline, histidine, arginine, cysteine, methionine. Now these are the 3, 4 or 5 amino acids which are very susceptible to free radical attack. They result in oxidative damage. We call them as protein carbonyls. And when a protein carbonyl is formed, they are fragmented. They help in cross-linking, aggregation can take place, and they become susceptible to proteolytic digestion. All these changes, oxidative damage to the macromolecules can cause increase in the flux of free calcium ions. So it will lead to cell swelling, permeability to sodium ions, which lead to further damage. So all these things are possible because of free radicals. Does it keep on happening? Then if it was keeping on happening, then we all would have been deceased by now. So there must be some mechanism in our body which prevents the attack of these free radicals, and they are called as antioxidants. So what is the cellular defense against reactive oxygen species? Remember, we cannot stop electron transport chain. We cannot stop macrophages from releasing. We cannot stop iron from releasing free radicals. We cannot stop that chain termination progression. We can find out how much is a free radical damage, that's all. But it is the body itself which needs to have certain mechanism, cellular defense against this reactive oxygen species. They are both a necessary and an evil. Necessary evil, what we say, are the free radicals. So what are the cellular defense against reactive oxygen species? So this process by which ROS are removed, are scavenged, and the substances which help in the scavenging and removal of these free radicals are called as antioxidants. So these are substances which at very low concentrations are also able to compete with other oxidizable substances, and they inhibit the oxidation of other substances. Basically, antioxidants are opposite to free radicals. Antioxidants are good for us. So let us look at the antioxidants. Antioxidants have been classified as preventive antioxidants. That is, they prevent the initial formation of free radical itself. In this, we have the category ceruloplasmin, which is a copper transporting protein, transferrin, which is responsible for iron transport. Thereby, it prevents the action of iron and further Fenton reaction, Hebowitz reactions are stopped. Ferritin, which stores iron. Albumin is also considered as an antioxidant. Myoglobin is also considered as an antioxidant. So let us look at the chain breaking antioxidants. They prevent the propagation of the free radical. Remember, one free radical forms another will form another. So this chain which is continuing is broken down by certain antioxidants. Now these include superoxide dismutase, glutathione peroxidase, vitamin E catalases, peroxidases. I'll talk about all this in detail. Now before we go further to antioxidants, these antioxidants have also been classified as enzymatic antioxidants and non-enzymatic. Remember, everywhere free radicals are produced, whether it is in the mitochondria, whether it is in the endoplasmic reticulum, or in the lysosome, or in the peroxisome, or in the nucleus. And everywhere the distribution of these antioxidants is necessary, and everywhere these enzymes are needed. So certain enzymes are present in certain locations to prevent ROS damage to that particular organelle. So we have the enzymatic antioxidants which are intracellular, include SOD, glutathione peroxidase, catalase, and glutathione reductase. Extracellular antioxidants include SOD and glutathione peroxidase, and the non-enzymatic antioxidants include the water soluble and lipid soluble. Water soluble is present within the cell and usually in the cytosol, includes glutathione, uric acid, and ascorbic acid or vitamin C. Lipid soluble includes the vitamin E, beta carotene and coenzyme Q. So let us look at superoxide dismutase. SOD is one of the enzymes which has been very important in superoxide dismutase. So we have oxygen and superoxide which will combine with one more to form hydrogen peroxide plus water. Hydrogen peroxide plus water. So this superoxide dismutase is responsible for superoxide anions. This is a primary antioxidant enzyme. It is present in the cell cytoplasm. It is a copper zinc enzyme, and in the mitochondria it is a manganese enzyme. It catalyzes the dismutation of superoxide anion. It is also present in an extracellular form of it, present in the plasma, lymph and synovial fluid. Catalase is for the of hydrogen peroxide. Hydrogen peroxide is for the H2O, water plus oxygen can be done by catalase. Catalase is important in peroxisomes, to some extent in cytosol or microsomes. Now one catalase in granulomatous cells, the enzyme protects the cell against its own respiratory burst. It decomposes hydrogen peroxide to water and oxygen. Glutathione peroxidase. Now glutathione peroxidase is present in the cytosol and mitochondria. There are two isoenzymes, one of which is selenium containing metal enzyme, and it catalyzes the reduction of hydrogen peroxide and lipid peroxide by reduced glutathione, and glutathione is oxidized to its oxidized form. I'll talk more about glutathione peroxidase and the other antioxidants in my next session. So let us continue with free radicals and antioxidants in my next session. So thank you.